Alrighty, thank you all for coming, and thank you very much to the conference organizers for inviting me to speak today. My name is Kelsey Moody, and I'm the CEO and founder of Ichor Life Sciences. And today I've been invited to speak a little bit about contract research in longevity and decentralized science. By way of financial disclosures, I'm one of those evil corporate types, so you can assume that everything that I'm talking about today I have some flavor of vested financial interest in. And I think it's important to think about the origins of our organization because I just finished my undergraduate degree when kind of the first wave of longevity-focused biotechs came on the scene. And it was really frustrating for me to see how one by one all these really promising organizations with very promising tech failed, and it was really demoralizing. And so when I set out to establish Ichor, I felt it was very important that if you're going to build a longevity-focused company, the company itself should have some longevity and not peter out after just a couple of years. So I started Ichor when I was in medical school in central New York, strategically located in my apartment next to Coleman's Irish Pub. And the real focus of the organization was how do we create a company that can advance longevity science, but in a way that we are not subject to short-term investor pressures, short-term thinking, and where we can create a situation where we can go after really aggressive, cutting-edge technology to move the envelope in this field. Fast forward 10 years, this year we celebrated our 10-year anniversary. And Ichor is now a vertically integrated pharmaceutical company with 3 locations, all based in New York State. We've got a footprint of about 30,000 square feet and over 50 people. And we operate a complete pipeline from the earliest stages of ideation and drug discovery through the clinic. And so with that kind of context, I wanted to share a little bit about current trends in contract research and how we're deploying Ichor as a discovery engine to accelerate and recognize the immense potential of this field through supporting startups, through academic collaborations, and through our own internal programs. Additionally, I want to focus a little bit on how these CRO trends present a very interesting and unique opportunity for DeSci to really advance novel technologies in a very different way than what's been done historically. So current trends in research, in the most reductionist form, if we want to build drugs, right? I'm a drug developer, our company develops drugs. If we want to develop drugs, you basically need 2 things. You need an understanding of the relevant basic biology, and with that understanding of basic biology, you have to identify and validate disease-modifying therapeutic targets. Now, this is challenging in a complex area of new science like longevity, because the relevant basic biology is still being understood. It's still being sorted, and disease-modifying therapeutic targets are still being validated. This is the definition of why there's so much risk in a novel space. So it's not surprising then that traditional drug development, especially in a new space, really begins with basic science at universities in academic laboratories. And if we look at the funding available for that, we have personal funding. So this is academic PIs raising grants and so forth from granting agencies, maybe accelerator programs, high net worth individuals and angels. And this system works really well in an academic situation because there's no time pressure. If I'm in academia and I want to spend my entire career studying a very obscure pathway that I'm passionate about, that's a thing I can do. Additionally, if we think about DeSci as a concept, there's not really an organization more decentralized than a university because you have operational freedom. You have the smallest autonomous unit possible, which is the principal investigator, surrounded with a small team of graduate students, postdocs, technicians, whatever, and those individuals can be deployed on very diverse research projects. Of course, as everyone knows, universities come with a caveat, which is limited funding and physical infrastructure resources as well. At some point, historically, big pharma was kind of the player that took over from universities and would translate basic science discoveries identified in academic labs and finance the development budgets, the clinical trial budgets, et cetera, to move from discovery into development and eventually a product. And I think for the longevity space in general, this distinction between discovery and development is really, really important. Discovery is understanding the basic science and validating the therapeutic target that you're trying to mess with in some valuable way. Development is taking early tool molecules that kind of establish proof of concept for that pathway and giving them the sorts of physiochemical properties that are necessary for them to be a viable drug. Development is expensive. It has its own challenges, but the risks are defined. For almost any sort of drug, I understand what sort of half-life, what sort of stability, what manufacturing stipulations need to exist in order for that drug to be viable on the market. Discovery is 100% risk. We don't even know if the basic science is correct or if the pathway is accurate. Now, what's been interesting is, so big pharma, obviously, because there's commercialization opportunities, also have larger cash outlays to be able to deploy on these problems. But like any other large organization, they tend to be bureaucratic, and you start being beholden to these time pressures, investors looking for returns, wondering what's going on, because there's a big dearth between when something gets started and when product comes out as far as pharmaceutical timelines. We all know this. Over time, big pharma has kind of transitioned more and more away from doing its own internal preclinical R&D, and even to a large extent, clinical R&D, and instead relying upon contract research organizations to fill the gap. I think the trend, if it continues, is going to be that big pharma basically sells drugs, maybe does late-stage clinical work, but everything else is largely outsourced to CROs. And with this model, we see kind of our traditional, what we think of as a startup biotech or pharma, which is going to be taking discoveries from universities, translating them through venture capital into assets that can be acquired by big pharma. But these entities exist as virtual companies where the majority of the work is contracted to CROs. What's emerging as a result of this trend is that CROs are actually a gatekeeper that's being positioned between university discoveries and big pharma acquisitions. And with this kind of framework, we've positioned Ichor to be a strategic partner for our various organizations that we engage with. Specifically, 3 of the pillars of Ichor. We have a discovery pillar, which is focused on target validation, basic science, does a lot of academic work with academic collaborators. We have a pharmacology group that's focused more on preclinical proof of concept, cell-based studies, in vivo efficacy studies. And then we have a clinical trial division that's looking into actual clinical trial design and the handoff and integration with big pharma. And collectively, these 3 pillars fall under Ichor Life Sciences. So when we started this organization, I honestly just wanted a company I could deploy on whatever the heck I thought was interesting, and we've successfully accomplished that. So now the question is, okay, of all the many things we could do with this engine that we've built, how can we deploy it to create the most value? And if we look at kind of the value chain of Ichor, it starts off with the fact that unlike most companies in the longevity space, we're a profitable organization. So we do contract research for big pharma, mid-tier pharma, and startup companies from ideation through clinic, and that allows us to own our own facilities. We own our own equipment. We have free cash to invest. And because we are strategically located in central New York in the middle of nowhere, it's very inexpensive to operate. I bought our first commercial building for 9 months of rent if I was in Boston or Silicon Valley. So there's certainly a value in being in a more affordable area. With these funds, we're able to establish and invest in more academic pre-investment stage projects. We've established, as far as I'm aware, the oldest aging Ph.D. program that's focused on drug development for age-related disease with our university partners, Clarkson University, SUNY ESF, and Syracuse University. And with this PhD program, we're able to apply industrial scale and budgets to academic problems without the time pressures associated with developing new technologies and new programs. I'd like to take a second and just share a few case studies to give you a scope of some of the projects that we're involved in. If you're in this audience and you have never heard of liquid-liquid phase separation before, please write that down and read what it is. In drug discovery, the canonical model is maybe like a ligand-receptor interaction, and all of the drug development work for, as far as traditional small molecule, is designed to disrupt the ligand-receptor interaction. In our capacity as a CRO, running hundreds of programs for hundreds of companies, all of the next generation, I can't say all, a large quantity of the next generation small molecule targets are proteins that do not undergo traditional ligand-receptor interaction. They undergo a different process called liquid-liquid phase separation. Phase separation is the mechanism through which most aging targets work. These transcription factors and all the results of these omics screens that you guys are hearing about typically follow a phase separation mechanism of action, and we're aware of at least a dozen programs in the aging space that have either failed or been significantly slowed because the organizations developing the tech believe that their drug follows a ligand receptor type of mechanism when it's actually phase separation. The challenge with phase separation is that it's a fairly new field from a biophysics perspective, and the sorts of high throughput screening assays that exist for conventional small molecule screens do not yet exist for phase separation reactions. With our colleagues in the physics department at Syracuse University and at Clarkson, we're building those. We've got a handful of publications out, and we've got a bunch in various stages of peer review, characterizing phase separation, their rates of reaction, and more importantly, how to drug them. If you have a target that's being very finicky and you're not sure how it's working or why, please talk to us. We see this all the time. We also have more conventional projects we're working on. This is one of our clients, Shipathesius, that recently published a biologic drug candidate with us that accelerates wound healing in diabetic and aged non-diabetic mammals. This product's about to be preparing for IND-enabling studies, and we're really excited about moving that into the clinic. And then we also are interested in new enabling technologies that might not necessarily be related to aging specifically, but can be supportive. This is a really cool diagnostic technology that we're developing. What this technology is a single molecule nanopore sensor. So the way most nanopore sensors work is you have a nanopore in a membrane, and you have some sort of analyte of interest that you want to detect that flows through the nanopore, and you're able to detect the changes in the nanopore's the membrane potential to identify what the analytes are. The problem with that kind of a technology is it's very difficult to engineer nanopores to have broad specificity for all kinds of different things you might want to measure. This technology is super cool. The way to think about it is our nanopore, instead of interacting inside of the nanopore, you can think of it as a drawstring bag where there's a floppy string that's outside of the nanopore, and we can engineer that string with antibodies or with antibody mimetics to bind any analyte of interest, and if the antibody or antibody mimetic gets bound, the nanopore closes, so we can engineer this to have specificity against any different target that you can engineer an antibody to bind. We've got a handful of Nature papers about this. For those of you that followed Theranos, this is basically their value proposition, but they kind of ruined the pitch for us, so we're developing this academically. We have other projects ongoing in phase separation, nanosensors, life extension, and also now cryobiology. If you guys are interested in any of those topics, we're around. I'd love to talk to you about collaboration opportunities and so forth. As these projects reach a certain level of maturity, we spin them out as IP holding portfolio companies of our organization. Couple case studies you guys might be familiar with because we've been working on them for a few years now. Lysoclear is an enzyme therapy for age-related macular degeneration, originally ideated by Aubrey. Over the last few years, we've identified a series of enzymes that can break down intraocular lipofuscin, which we believe is the causative agent of the disease. And we've shown that these enzymes are efficacious in cell-free, cell-based, and in vivo assays. We also developed with our portfolio company, Octus Biologics, an orally bioavailable antibiomimetic platform. So this is a binding scaffold that can bind therapeutic targets similar to antibodies, but unlike antibodies, they're stable at room temperature. They can be expressed readily in E. coli, and because of their stability, we're able to engineer them to be orally bioavailable instead of requiring infusion. And we're going after all of the canonical antibody targets with this technology. An offshoot of this technology, SPR, surface plasmon resonance, is the gold standard technology for drug target binding in industry. And we're going so far as to not just use SPR in our workflows, but we're developing next generation regeneratable SPR chips, and we have a grant from NSF to work on that. I should also mention, outside of our companies, we are actively invested in external companies. I've got a few of them listed here, and in the last 18 months, I want to say we've done maybe 1.5 million or so in placements in new organizations. So if you have a new technology that you're interested in turning into a company, we'd like to talk to you about that as well. How does an organization that is a service provider in the middle of nowhere in Syracuse build the relationships necessary to raise venture capital and to position their assets for acquisition? Enter the CRO. We've been doing the contract work for a while, and we're really partners with the organizations that we engage with. We're at the stage where life science VC come to us with projects that have not even been incorporated yet to run the full preclinical programs before they even spin them out into a company. We have pharmaceutical partners that sit down with us and outline the next 3 to 5 years of their strategic priorities, and we're at the table having active conversations to help them to achieve those goals with what they have in their pipelines. But that also gives us a unique opportunity to take things in our own pipeline and present them to these partners when there's a good fit. And we think that this is going to allow us to be really competitive for facilitating deals and acquisitions for our portfolio over time. There are some implications, I think, from this whole idea that pharma is outsourcing more and more, and you have this emergence of contract research organizations that's really important for the whole movement of decentralized science. I won't spend a whole lot of time on it because other people have talked about this and other people will continue to talk about it, but I love this concept of an IP NFT as a novel mechanism for funding science where people can invest in a token and receive intellectual property as a result of that, and that can be used to facilitate and accelerate research. I do think the applications of IP NFTs are going to be super powerful for general technology development and also for pipeline development. I would caution, just in our experience with working with academic spinouts, I saw a lot of stuff when we were looking at IP NFTs about interfacing with tech transfer offices and spinning out therapeutic assets. Our experience with rare exception is that therapeutic assets tend to be very weak when coming out of tech transfer offices, and there's very few examples, at least with the companies that we interact with, where we've seen the relevant IP that eventually goes to market come out of tech transfer. Usually these things are developed later. The IP usually covers tool molecules, but not what actually becomes the eventual drug. Regardless, we think at least that this is a really exciting area for commercialization and for funding. And I like the idea of using DAOs and individuals and syndicates in this kind of ecosystem because there's such an outsourcing from pharma to contract research organizations, you can find a contract research organization that can do literally anything you could possibly imagine you would want to do. What's nice is if you outsource to CROs, you can avoid university overhead, which can be up to 75% on the dollar. Academics who have all of the knowledge around the particular projects can drive them really quickly, and you can leverage CRO service offerings, speed, and scale. As just an example, we had one academic that we collaborated with, really interesting target of interest. It would have taken their grad student 5 years to make enough recombinant protein to study their mechanism of interest at 1 liter bioreactors per run. We put on 100-liter bioreactors and we made the proteins they needed for their studies in a couple of weeks. That sort of scale can be applied to translational projects and new tech as well. I do want to share, this wasn't actually through an IP NFT, but I think this one case study, which will wrap up my talk, really illustrates the power of how one can deploy CROs quickly and strategically on diverse projects. So we had a high net worth individual engage with us who was diagnosed with idiopathic pulmonary hypertension. This is a progressive disease that underlies or that affects the precapillary pulmonary vasculature. Basically what happens is the blood vessels between the lungs and the heart start to constrict, and that leads to an excess load on the heart, which eventually kills the patient in 2 to 3 years if untreated. This patient was enrolled for a clinical trial for APD-811, which is a drug that's supposed to be effective against the disease. This was a phase 3 clinical trial. Correctly so, the patient was concerned as to whether they're in the therapeutic or the treatment group. So the first thing they did is they gave us one of the pills from their trial and we sent it out for mass spec. And we were tasked with putting together all of the IND documentation and clinical documentation around this drug. While we were waiting for the mass spec data to come back, we outlined an entire program for sourcing API, formulating, doing all of the stability testing that had already been demonstrated, cell-based studies, and also in vivo studies that would be required to recapitulate the entirety of the IND package, non-GLP, for this client so that they would have a research-grade product to take if they couldn't get the clinical product. The total cost to do that, had we executed, would have been on the order of $250 to $500K, something that a syndicate or a high net worth individual could certainly afford to do. Now, a lot of people have very firm opinions on FDA and offshoring and right to access. I'm going to intentionally not have opinions on any of these topics. I just think it's kind of interesting that an individual can come to an organization like ours, have a workup like this in a couple of weeks. It turned out that they were in fact on the clinical trial drug, so we didn't proceed with the program. But that's something that exists and is accessible to anyone that has those sorts of resources. So when I'm looking at IPNFTs and DAOs and investor syndicates and so forth, and the availability of CROs that have deep expertise in very nuanceable technical areas, I think that there's a lot of potential to really disrupt how technologies are developed based on these business structures. So in summary, CROs offer the stability of an established company with, as I hope I've demonstrated, the upswing potential of a therapeutic startup. Ichor itself is actively looking for partnership opportunities. We do academic research. I've got Ph.D. students that need to publish. We're really interested in advancing academic science and leveraging our industrial muscle when we can. So if you guys have projects you want to collaborate on, we're super interested in that. We're certainly recruiting prospective graduate students. And obviously we do work for hire, but that's not why I'm here today. Additionally, we think that DAOs and IPNFTs offer unique models for advancing scientific research, and through the use of strategic CROs can really function outside of traditional academic systems. And with that, I don't know if we have any time, but otherwise I'm available for questions now or after. Thank you.